First, thanks to the organizers for really putting together extremely stimulating workshop. So excited to tell you a bit about my work today. I've stolen some quotes, this one from Feynman and one from Yogi Berra, really capturing the idea that in biology, there's a lot that we can really learn just by looking carefully at biological systems. And I think we're at a really exciting point, in this post-genomic era, where we can learn a lot, especially from considering and reconsidering, in many cases, all that biological diversity has to teach us. So, the kind of thing that really animates my work is stuff like this. What you're watching here is not a worm, but just one cell that is hunted down, attacked, captured, and is now in the process of devouring what's actually an animal. So cells are capable of some of these behaviors of, I think, sort of remarkable sophistication. And we know a lot about cells, but I contend that we don't really know a lot about how cells can do stuff like this, much less predict it from a genome. And so the core question, the motivation of my lab's work is to understand how cells control complex behaviors. And so a lot of times we start by carefully, quantitatively characterizing cell behavior, often in terms of cell shape and movement patterns. We poke at the cells a bit, using often a combination of theory and experiments to get at mechanistic underpinnings. But we want to understand not only what cells are doing in the lab, but what they're doing in environmental context. So we really try to focus on naturalistic behaviors. And we want to put that all together in sort of an integrative fashion to draw comparisons that allow us to understand something about how cell behavior evolves. And so if, like me, you're interested in general principles, if you look out into the world at all sorts of weird and wonderful cells, you're admittedly confronted with a bit of a challenge. So cells come in all these different shapes, sizes, and abilities. They hunt things, as you saw. They also sometimes, maybe hold out or hang out holding hands and do other inscrutable things. But the diversity presents us with opportunity. This is a tree of eukaryotes. There's a ton of diversity of form and function. There's some hope in that despite all this diversity of form and function, these cells do have shared ancestry, so depicted in this convergence of all these branches here. So a lot of the components that these cells are built out of are kind of similar, the same in many cases. Cells are also often similar sizes, so they're subject to similar kinds of physical constraints. And finally, we have this opportunity to select among or across all this diversity to find systems that highlight, through their unique characteristics, particular biological features that help us powerfully answer questions. And so the talk that I'm going to give today, we're going to hop a bit around the tree of life. We try to weave together these different stories with some themes that I think are consistent with the aims of this program, this workshop. So what I want to convince you of ultimately, though, if nothing else, is that whatever biological question you have, there's a protist out there for you, and I hope you find it. Okay, so our first story is actually going to take us, it looks like this laser pointer isn't working so well, but anyway, to this branch that's near us, where we live. So animals are right over here, and we're going to look at some critters called choanoflagellates. And it turns out they're very useful for addressing this question of how animals first evolved from unicellular ancestors. There's this major evolutionary transition from unicellular life to the complex multicellularity that we see in animals. It's hard to study this using something like paleontological methods. These animals didn't leave too many fossils. But what we can do is look to the choanoflagellates, which are in fact the closest living relatives of all animals. And the idea is we compare and contrast their biology with that of animals to understand something about their last common ancestor looked like, to ultimately understand how multicellularity arose. So I started thinking about this sort of thing as a PhD student in Nicole King's lab, who was and continues to be a fantastic mentor. And the first time I met with her, we sat down and she showed me this sort of video. Okay, so this is a choanoflagellate cell body here. This is part of a structure called the collar complex. There's a flagellum here. The cell's going to beat this flagellum, pull in a bunch of bacterial prey. And it turns out that under certain environmental conditions, these cells can feast on bacteria, grow and divide, and grow into these three-dimensional rosette structures. So this is colony development, multicellular development, in a unicellular relative of animals. And so when I first saw this, Nicole told me, okay, there's probably something biophysics-y here. I have background in physics. Okay, you can work on this problem. But so I really wanted to understand how this thing worked, how they build this structure. And the first thing that I set out to do is just carefully look at the shapes of this structure. So to understand how this thing is built, I want to know what it really looks like. And so what I did is I imaged a bunch of cells by confocal microscopy. And so these are just three orthogonal views of the same structure in each column here. So I can give a pseudo time course where I have from 4 to 12 cells, the different shapes of these structures. And So I wrestled with this problem a little bit, but I made one plot at one point where what we're looking at is the rosette width and volume and how those scale relative to one another on a log-log plot. So it goes from 4, 5, 6, 7 up to 12 cells here for all these data points. And what I saw was when you get between the 7 and 8 cell stage, there's this growth transition. And we go from something with a large scaling exponent to something that's about a scaling exponent of 3. Okay, and so if radius scales like, or volume scales like radius cubed, right, that's sort of an isotropic-like expansion. And so the picture that I kind of had in my mind is like if you blow up a balloon, dramatic shape change that happens early, and then you hit this swelling sort of phase. And so that got me thinking about, okay, is there sort of a physical explanation for this growth transition? And the idea that I kind of came up with is that it turns out these cells are held together by this little extracellular matrix that we can see. We know this extracellular matrix is important for colony formation. And so the physical picture is, okay, they secrete this limiting little blob of ECM. Cells grow, push on one another, and they're constrained in their packing. And at this 8-cell stage, maybe they hit this jamming-like transition where they're all smushed together. And so the only way for the structures to grow is for the cells to all push on one another, giving this sort of inflation. And in part because of this workshop, I wanted to call out something that was really important for the development of this idea. And that was hanging out and trying to explain the biological problem I was working on to some really great theorists. So this was during my time at Berkeley. George Oster, phenomenal theorist in biophysics, and Oskar Halachek was there at the time too, also a theorist who thought about cell mechanics. And it was really sort of the process of trying to explain this problem to them that helped me develop this physical intuition here. Okay. And so I did a bunch of experiments, and it turned out that, this physical picture was actually consistent with what we saw going on. There was this increased stress in the colony as they passed this 8-cell stage. Okay, and I wanted to push this idea a little bit farther. So I teamed up with the Mahadevan group, in particular Teresa, to build simulations based on this simple biophysical model of colony formation. And so there are these 3 parameters that describe this model. There's one parameter that's the cell shape, so how elongated the cell is. Another parameter that describes the amount of extracellular matrix relative to the amount of cells. And then a final one that describes how stiff, how deformable this ECM is relative to the cells. And so what we could do with these simulations is mess with those parameters and look at what kind of morphologies emerge. And what I was really happy to find is that not only in parameter space do you get a region of rosettes over here, right? But I get this diverse range of different sort of colonial morphologies. And what's even more exciting here is that this actually maps onto diversity of forms that you see in nature. So we have this sort of simple physical picture. The idea is there's this physical constraint that gives rise to this robust morphogenetic capacity. And so one thing that's not in the model is anything specifying the details and orientation and order of cell divisions. So this is a robust morphogenetic process where you don't have to tightly couple and control cell divisions that will just give you these reproducible morphologies based on this physical constraint. Okay, and what's even more fun to me is that as the choanoflagellate community grows and more people are working on these organisms, we increasingly have genetic tools. So here's an example from Thibaut Brunet's lab where they made a mutant in part of what's called the hippo pathway, this a pathway that's really important in animals for controlling tissue growth and organ size scaling. And they see this interesting rosette defect where you get these bigger rosettes that tend to have this disordered extracellular matrix that really maps on quite well preliminarily to some of the predictions that we made from this sort of model. And so I hope that this kind of physical picture can continue to be useful in helping us interpret results of perturbative experiments within choanoflagellates. And so we're starting to think more about rosette size and shape control and colony control more broadly across choanoflagellate diversity. Okay, so takeaway here, right, is the importance of physical constraints in conferring robust functional outcomes in the face of stochastic variability underlying the dynamics of, in this case, cells. Okay, right, and so this might be part of a broader theme where we see across diverse lineages in animals and bacteria and other sorts of protists, where this basal extracellular matrix is sculpting multicellular morphology. So I think it's sort of maybe an interesting evo-devo principle where there are a lot of questions that you can start to ask when you're applying this sort of framework to explaining diversity of form across systems. Okay, so that was a prelude, story 1, to our next story, which is going to take us to a very different region of genotype and phenotype space, and that's over here to the ciliates. Okay, So here we have what's really the main model system in our lab at the moment. This is a unicellular organism, more or less as I first encountered it in a field sample. These things have fascinated protistologists for about 150 years. This is a cell that walks across surfaces using leg-like appendages with this uncannily insect or maybe robot-like behavior. And that really, when I first started to see these things in field samples and figured out that it is in fact just one cell, I wanted to understand how it is that a cell can control all this apparently sophisticated, complicated movement without a nervous system, without a brain of any kind. And so this is just another view of a cell. Cell's about 150 microns long. You're watching it run around in a high-speed video recording on a cover slip. And there's all this apparent sophistication of leg movement. So all the cell movement in this case is driven by movement of the leg-like appendages. And what I wanted to understand is really what the nature of coordination among those appendages are, and then how that might actually be controlled. So here is an admittedly very information-dense diagram. So I just want you to take away a few things. So this is a representation of the stepping patterns of cells. So each one of these nodes on this graph is postures or configurations of which legs are moving and not moving. And these arrows represent transition probability, so how likely the cell is to go from one set of legs moving to another set of legs moving. And there's a lot of variability in the stepping patterns of the cell, but nevertheless, there's this overall sequential logic to what the cell is doing. And so we're finding functionally significant gate state transitions here. But what I want you to take away is that these kinds of representations, I think, are generally quite useful in interpreting the effects of perturbations. So as we start to poke at control modules in the cell, the way that the gate is affected in this kind of representation can give us insight into the nature of these control modules. But I think these pictures are also useful in drawing inference and making predictions about cell behavior. And so I'm going to talk a little bit about that in this next part of the talk. If you want to know more about the details of this work, check out this paper. That's all I'm going to say about this work.
Track, so,
yeah.
Very briefly though.
Yeah, please.
Eigenworm style like tracking of the limbs, and then you're like,
yep.
Shit.
Exactly. Yeah, exactly. And we're building a lot on that moving forward in the lab, and I'd be happy to talk more about that later. Okay, right? So I said I think that these are, it's a general way of coarse graining the dynamical complexity of biological systems to draw inference and make predictions about biological systems. So one way we've been pushing that idea is to maybe make the most extreme transition possible here from ciliates to the amoeba, which are definitionally the most morphologically complex sort of cells that you can find, in that they're defined by not having a defined morphology, right? Okay. And so this amoeba is so crazy that they actually named the genus Chaos. So this is a phagocytic pseudopod of one of these giant amoeba. The whole amoeba itself is millimeters long, and it's right now attacking this prey item here. This is a paramecium that's about 250 microns long, and this is a phagocytic pseudopod where it's devouring this paramecium. And so I spent a lot of time watching these amoeba hunting, and I got the sense that I could sort of tell just by looking at an amoeba, watching it crawl a little bit, that it was in this hungry state where it's going to pounce on prey, versus amoebae that were just sort of exploring their environment, insensitive to prey stimuli. And I'm not the first person to make this sort of observation. I'm not crazy. Jennings in the early 1900s also saw these sequential but admittedly complicated and variable types of amoeboid movements, and he also described these sort of hunting patterns of the amoeba. Okay, and so what we can do is segment these amoeba, so getting all these shapes in different time points of the video, and look at those shapes. And so these are just two amoeba. That one in the middle is hunting, that other one was sort of migrating here. And the question is, can we infer whether a cell is hungry, hunting, attacking, versus migrating just by looking at the movement patterns of these things and not actually watching them eat things? And so we coarse grain all of those shapes. So here we just use PCA to get these postures of amoeba, if you want to think about that the amoeba return to over and over again as they crawl around on a cover slip. So there are about 5 of them in this case. And when we start to look at transitions among these different postures, it turns out that there are these diagnostic sort of hunting transitions that we find that clearly distinguish hunting amoeba from these migrating amoeba. So they adopt similar kinds of postures, but it's really about the sequential logic of those shape transitions that picks out hunting versus migrating. And I think that's what my eye is picking up on when I watch these things. And so first PhD student in the lab, Christina has bravely picked up this kind of, or this project, this morphodynamic analysis and cell state inference project, and she has taken it, I think, in a really exciting new direction. And that is to another branch on the tree of life, back toward our quinoflagellates, into animals. And so, to my mind, the most protist-like of animal cells has got to be immune cells. So this is a macrophage from, it's a bone marrow-derived mouse macrophage. There it's hunting, eating some schmutz on the cover slip. And so Christina has been doing work to see if we can watch macrophages crawling around in a cover slip and make inference about cell state from those morphodynamic transitions in a similar fashion. So Christina is a brilliant experimentalist, and she came to the lab with relatively little computational background, and she's been able to develop this deep learning-based image analysis pipeline to analyze all sorts of shape and movement patterns in amoeboid cells, and has got some really exciting preliminary results where we think that we may, in fact, be able to, based on shape and movement patterns of these amoeba, decode the inflammation state of these cells, right? So we can do this, a similar sort of cell state inference to what we did with amoebae, but in this case, in mammalian cells, okay? And so I'm really excited about this work. Stay tuned for more to come from that. And if you want to learn more about it, go talk to Christina, she's here. Okay, so our final, I guess, prelude to this talk is going to take us back to our ciliates once again, okay? So I mentioned that something I'm very interested in is understanding how cellular behavior evolves. And I think that euplodes, this walking cell that I mentioned, gives us quite a powerful window onto that question. And that is in that we have a lot of diversity of form and function among these cells, but they stick to a kind of similar sort of body plan. Okay, so these are just trajectories of 3 different species of euplodes cells running around in the environment, and these are microtubule cytoskeletons of these cells. And so one thing that we have learned about euplodes is that this microtubule cytoskeleton, these are these structures that are associated with those little leg-like appendages, we think are important for mechanically mediating coordination among the appendages. So we have this interesting sort of conservation, but also slight variation on this architectural theme, and we think we can use this biological diversity to draw inference, or to actually learn rather, about how cell behavior evolves through modification of cell geometry. And the most extreme version of that sort of process, I think, comes from a wonderful organism that I'd like to tell you about in the last 5 minutes here, which is one that I found out in the field. So I've been very lucky in that I've gotten to do some field work in the island of Curacao. And we've explored the diversity, all around this island. I've sampled all sorts of different environments. But one place that I hadn't sampled was the diversity within the lab and in Curacao at the field station. And so I looked to this aquarium filter, and I found these wonderful cells residing on that aquarium filter. It turned out that sampling cells from an aquarium filter is a pretty good idea if you want a cell that's going to be well behaved in the lab, because these things live on trash in stagnant environments, which is exactly what an incubator is. And so I was able to isolate this culture of cells, happened to be euplodes, from this filter. And after a while, I started to notice this. So I picked, clonal isolates to establish cultures, and I noticed all this morphological variation. And it turns out that in addition to being, really, really big, these cells are very different behaviorally. Okay, so this is all a clonal population of cells. These are all euplodes cells. We've got this big guy in the middle and all these little guys around the side. And if you watch these carefully, what you will see is they're fearsome cannibalistic predators. So they exhibit this dramatic phenotypic transition where they get huge and cannibalistic. So we have this associated morphological behavioral kind of transition in the cells. So here's some electron microscopy to show you that there are these supergiant cannibals. There are these normal morphs, that's sort of the standard euplodes cell. And there are also these other interesting morphologies that we don't really know too much about that have this apparently sort of defensive morphology where they're too big to fit in the mouth of the predators. All right. So there's a lot of interesting behavior and morphological diversity in these things. This is just to show you, if you look at the movement patterns of normal morphs versus supergiants, they're, qualitatively, obviously quite different. So these normal morphs are running in straight lines. These little curly things are swimming trajectories. These supergiants are running in weird looping trajectories a lot of the time, and they really don't seem to spontaneously swim much at all. So I wondered if there might be a behavioral trade-off associated with supergiant formation. So this is a mix of supergiant and normal cells. And in these experiments, I'm just disturbing them from a cover slip, so shaking the cover slip or pipetting. And if you look, you'll see these supergiants really sort of seem to mostly tumble helplessly when they lose contact with the surface, whereas the normal morphs, I don't know, there's one swimming over here, swim quite well. So these are just trajectories of these normal morphs swimming versus the supergiants swimming. They seem to have this sort of limited dispersal capability. Yeah.
You know they're the same species.
Because it's from a clonal isolate.
Okay.
Yeah, and we have a bunch of sequencing on these two. So we have transcriptomes for all the different morphs of the cells as well. And so, yeah, it's from a clonal isolate. That's a good question, though. Okay, so there's this behavioral trade-off. And one last kind of point that I want to make. So I'm trying to motivate you all to work on protists. But I need to help you a little bit because they can be admittedly challenging to work on. So one thing that will really help you is to fine-tune your culture conditions. And that turned out to be a breakthrough in being able to actually study this really interesting cell phenomenon in the lab. So I mentioned, when I had isolated these cultures, I would spontaneously see something like this. Oh, there's a supergiant in there. I'm going to watch it, see what I can learn about it. But I had trouble keeping these supergiants around in the lab. And it wasn't until really carefully, and this is in part assisted by some fantastic undergrads assisting with this work, carefully dialing in the nutrients within the environment that I was able to reliably get supergiants. And I learned something even more interesting. So we were trying to figure out, what kind of induced the supergiants to form. And one thing that we found quite robustly is that what seems to be required is a relatively recent phase of rapid growth. So these are just growth curves. So the population grows up. This is just all the cells. And right as we hit stationary phase, we see the emergence of these supergiant cells that, pop up and then persist for a while and then kind of go away. But this happens under specific nutrient conditions. So this is no added nutrients, just seawater dilutes seawater, no growth, no supergiants form. But there's this sort of Goldilocks regime of moderate nutrients where you get nice cell growth and the supergiants form pretty reliably. But if you dial up the nutrients too much, so there's a lot of extra bacteria around in these cultures, we seem to sort of suppress the supergiants. And so there's a lot more to learn about this, but there's this interesting ability of these cells to continue producing supergiants through these cell divisions or revert back to the normal morphs to these asymmetric cell divisions. So there's this fine-tuning of morphology and behavior to specific environmental conditions. I'm running out of time, so I'm just going to quickly pull us to the end here. So if you want to learn more about this, check out this preprint. But there's this fantastic, interesting phenotypic transition that happens under very specific environmental conditions that's associated with sort of behavioral trade-offs. And there's, I think, a lot more to learn about this system. But what I think it really vividly illustrates to me is this idea here. So if you want to understand, I think like I do, the evolution of cellular behavior, what we really have to understand now is the evolution of cellular geometry and morphology, right? So among ciliates, this is just a depiction of ciliate diversity. There's this phenomenal diversity of form and function and size in these different cells. But despite all of that variability, synapomorphy, so a shared characteristic of all of these ciliates, is a particular kind of molecular and organelle-based cellular architecture where they have these ciliary arrays that are anchored into the cytoskeleton. And it's really sort of rearranging that cell geometry that gives rise to this functional diversity in these cells. And so I think they are really a vivid illustration of something that's more generally a principle of the evolution of cell behavior, that it's really about the emergence of new types of cytoskeletal organization and cellular forms that give cells to access of new types of phenotype space. And so I think for us to really make progress on this question of the evolution of cellular behavior, what we have to understand is cellular development and really build up our understanding of evolutionary developmental biology of unicellular organisms. So that's, I guess, my hypothesis to lob out there. And I think there's a lot to learn. And I think that also, this is a great opportunity for bringing mathematics and biology together, where I think part of the story that's going to be really important is an appreciation for the kinds of physical constraints that can for robustness to these morphogenetic processes. And we really don't know a lot about how those morphogenetic processes are controlled. So a lot to learn, a lot of open questions, more questions than answers. And with that, I'd like to thank all sorts of folks who just absolutely make it a joy to do science with every day, lab mates and collaborators, and also to thank you for your time and attention. Question? Yeah.
Rosettes, what kind of ECM is in the middle? And does it have any kind of unique structure that might affect its like mechanical ability to kind of hold?
Great question. We don't know a lot about that. So the main component that we know that is in that ECM is there's a protein called rosetteless. So it's a C-type lectin that probably mediates really, adhesion to this ECM, glycoproteins, all sorts of stuff in there. There's some preliminary evidence that suggests there are proteins that have collagen-like repeats in there, so maybe a little collagen-like. But I would love to know more about the composition of ECM. And there are of course, many other people who've sort of landed on this idea. But I think extracellular matrix evolution is a really important aspect of the evolution of animal multicellularity. And from my perspective, I like to think that the unique mechanical properties of animal ECM might have actually, made possible these large flexible body plans that really, I think that's really the main unique characteristic of animal biology, is that animals can get really big and move around without ripping themselves apart. And I think the ECM is a really important part of that. Yeah.
So this the leg is cilia.
Yeah, I didn't talk about this at all, but yeah, bundles of cilia, yeah.
They do the metachronal wave?
They don't. Distinctly not metachronal.
Okay,
Yeah.
So then how do they move back and forth?
Yep.
Okay, they change the direction of the power stroke and recovery stroke?
Yes. So we're trying to work out that in the lab right now. So we've just got this great ability now, and undergrad actually led this project where we can get automated tracking of all the appendage movements. And so from work from others as well has shown that there is electrical, bioelectric signaling that controls something about the type of waveform of these different cilia. How that's coordinated across the body is not really known. But yeah, the legs can execute different types of beat patterns. And so there are distinct groups of cilia that are actuated in a very specific way to get that reversal of the movement of the cell. And then there are other complicated waveforms that we see the cell execute. So when they're walking forward, you tend to see this sort of recovery stroke, power stroke, sort of swimming like this. When they shoot backwards, there's waving like this from the front legs and sort of a shrimp-like movement in the back one. So there seems to be quite a lot of differential control of these different populations of cilia in ways that are a bit hard to understand, actually, admittedly. I guess I saw your hand first.
I have another question about the rosette.
Yeah.
So the ECM, is this something that the cell secretes on its own when it?
So we don't know exactly whether there is control, like differential control of ECM deposition across the rosette, but the model is simply that each cell has a set amount that it's secreting and then it stops.
I see. So I was wondering if the thing can grow in multiple layers of cells, so that it grows even larger.
Yes.
Because I feel like if it's only one core, then the maximum size is sort of limited. So eventually you'll hit some type of plateau because the cell space.
Yep. So we never see multi-layered rosettes, but what you do see in species that grow especially large rosettes is you start to get these lobes on the rosette and they get a bit lumpier. But one thing that this model, the simple simulation cannot account for actually is really big rosettes that are completely closed, but also have this, lumen-like space in the middle. And so I don't have a good understanding of how that works, but it is the case that you never get these multi-layered structures. And that's partly because of the cell polarity and that the polarity of divisions is maintained. So, there's no obvious pattern to the details of the orientation of the division plane, but it's always along that apical basal axis. So that is part of, I think, what gives you this, single layer morphology. But with these really big rosettes, I think there does have to be some additional feedback or control on ECM secretion that is not accounted for in my simple model. Yeah. So
when you're introducing new protists to your lab, do you have a philosophy about when or whether you start trying to build genetic tools or start trying to apply drugs that maybe inhibit molecules of organisms?
That's a really great question.
Watching.
Yep, exactly. Yes. And so I didn't talk so much about, mechanistic studies here, and that's absolutely a major emphasis of the lab as well. So there's not a single sort of succinct answer to your question, but I think the general philosophy is, there are infinitely interesting organisms to work on. So some things that we need to really have an organism that we're going to invest in is we need to be able to culture this thing, to get at least hundreds of thousands of cells per mil usually, or something like this, so that we can actually run the basic experiments that we want. So we've got to dial in culture conditions enough to give us reproducible growth, stable growth, and these relatively high densities. So that's one thing that really enables us to do these experiments. In terms of investing in molecular tools, I'm still trying to figure that out. So it's a challenge. One thing that helps is if there is a broader community of researchers who are also interested in the organism, especially from different kinds of perspectives. And so that's something that's really helped with choanoflagellate biology. Okay, they're for understanding animal origins, but there are a lot of different facets of that question. And there's a community of researchers who work with these organisms and together are developing tools. And so I think having sort of a critical mass of interest would help me feel better about the development, really investing in the development of genetic tools, because then you're serving a broader community. It's not just, okay, it's only our lab that's going to work on this. So I think, the unique biological features of the system itself have to give you some really powerful insight into a fundamental biological question. So if you could study it in yeast, or if you could study it in C. elegans, 100% do that instead. So it has to be like, this is the only way you're going to answer this really interesting question. So I think that's the other really key feature of non-traditional systems. Now, how you make that call, whether it's interesting enough, I don't really know, but because like every organism is weird and wonderful in its own ways. The other small piece that I want to add, sorry, this is a long-winded answer to your question, but some organisms just seem to be much more manipulable than others. And so I think it does pay to consider some diversity. If you have, some features of a system and you can find multiple different species, having that diversity can help you, find the one that's more transfectable for whatever unknowable reason. So that helps as well. Yeah.
Thank you, Ben.